In this video, we are going to have a short tutorial on DNA structure and also the mechanism of its formation. We will also look at the main part of a DNA molecule and their location in the human body. Don't forget to subscribe after watching so that you can receive every update from this channel. So you have these objectives for today's tutorial. We are going to describe the three main parts of a nucleotide. We will also describe the DNA molecule as a whole and also demonstrate the complementary base pairing that exists in the DNA molecule. So what then is DNA? The term DNA stands for deoxyribonucleic acid. So it's just an acronym. It's just an acronym which stands for deoxyribonucleic acid. DNA, as we know, is a genetic material because it helps in the transmission of traits from parents to offsprings. DNA is a main molecule which helps in the control of protein synthesis in the body because it has sites on its structure we call the genes. So these genes are special regions on the DNA that code for particular protein. So if you have a protein A, it has a particular region on a DNA molecule which enables it to be synthesized. And that region on the DNA which enables the proteins to be synthesized are what we call the gene. So basically it controls the whole body because the body is made of proteins and DNA controls protein synthesis because of the location of the genes which are found on the DNA molecule. So why then is DNA important? As we said earlier, DNA is enabled or suitable for the transmission of genetic traits or information from parents to offsprings. These traits are all controlled by DNA because we have sites on the DNA we call genes which enables these traits to be synthesized. When there's transmission of traits from parents to offsprings, there is what we call DNA replication, and this replication comes with small error. That's why DNA is important. Also, we can't live without DNA molecule because, as I said earlier, DNA controls protein synthesis in the body, and the body is basically made of proteins. From the muscles back to the blood is made of proteins. And all these proteins are synthesized by particular genes located on the protein, on the DNA. So we can't do our DNA. It's very important because we can't live without it. So let's look at the DNA structure as a whole. So this is the structure of a DNA, and it is a double helical structure made of two strands. So this is a strand 1 and this is a strand 2. These strands are bonded by hydrogen bonds and later intertwined into a helical structure. When you take one strand, that one strand is a polymer that is composed of several monomer units, and this monomer unit which constitute one strand of a DNA is what we call a nucleotide. So basically these main components are what is found in a DNA molecule. This strand is a polymer made of several residues of monomer units which we call nucleotides. So when you take this one, you have several of these nucleotides in there. So you have three main parts of the DNA molecule because it composed of several nucleotides. We have what we call the nitrogenous bases. We are going to look at them. We have deoxyribose sugar and a phosphate group. These are nucleotides or monomer units which are used in the synthesis of the polymer which forms one strand of a DNA molecule. The nitrogenous base, the deoxyribose sugar, and the phosphate group. The nitrogenous base is bonded to the first carbon of the ribose sugar. Then the phosphate is bonded to the fifth carbon of the ribose sugar. Of course, it is called deoxyribose because the carbon 2 has no hydroxy group. So it is referred to as deoxyribose sugar. The carbon 2 has no hydroxy group. That's why it is called a deoxyribose sugar. So let's look at how basically the monomer units DNA are bonded in polymerization to form one backbone or one strand of the DNA. So So as I said earlier, this is a monomer unit which are bonded sequentially to form a polymer which forms one backbone or one strand of the DNA molecule. So we have one nucleotide. It has the nitrogenous base, the ribose sugar, deoxyribose sugar, and the phosphate group. Now this phosphate group is bonded to the fifth carbon of the deoxyribose sugar by a bond we call a phosphoester bond. So this is the third carbon which has a hydroxy group. The third carbon on the sugar has a hydroxy group which is bonded to the phosphate of the next nucleotide, and the phosphate also bonds to the fifth carbon of the next sugar. Now this phosphate has one ester group with this sugar and another ester group with this sugar. This means that when we have two nucleotides, the bond between them is two esters. So the bond that initiates or that joins one nucleotide to the other in a DNA molecule or in a DNA strand is what we call a phosphodiester bond. A phosphodiester bond is diester because this phosphate has one ester group with a hydroxy on the third carbon of the top sugar and another ester with a hydroxy on the fifth carbon of the next sugar. So you have two esters by phosphate. So it's called a phosphodiester bond. Series of the phosphodiester bond are initiated from the third carbon to the fifth carbon sequentially to form this polymerization, which we call a nucleotide, one strand of the DNA molecule. So as you saw earlier, when we take just one strand of these are formed by polymerization of several residues of the nucleotides. So taking this one, the whole of this is just one strand of the DNA molecule. So to form the DNA molecule, two of these strands come and bond to form two strands. And the bond is initiated between the nitrogenous bases by a rule called complementary base pairing. We are going to look at them in the few slides ahead. So that's how basically DNA structure looks like. Now let's come to nitrogenous bases. As we said earlier, these groups are the nitrogenous bases. So we have one nitrogenous base in each nucleotide, which enables two strands of DNA to bond to form that helical structure. We have two main groups of nitrogenous bases. We have what we call purines and pyrimidines. And the purines we have adenine and guanine, and for the pyrimidines we have thymine and cytosine in DNA. Looking at this structure, the purines are composed of double ring, but the pyrimidines are composed of just single ring. As you can see, they are all heterocyclic rings because they have different atoms in the ring, but the purines are all double ringed, whereas the pyrimidines are all single ring. We can also look at them in terms of the number of atom hydrogens in the ring. This one has 4 different inner hydrogens in the polyheterocyclic ring. This one has only 2 nitrogens in the heterocyclic ring. So this ones have 4 nitrogens in the heterocyclic ring. This one has 2 nitrogens in the heterocyclic ring. This one is double rings composed of one 6-membered ring, a hexa, and one 5-membered ring, a penta. But this one is composed of just a single ring. They are all heterocycles, of course, but this one is just a hexa ring. So the purines and the pyrimidines, they form the hydrogen bonds, as we said earlier, in the DNA molecules to initiate 2 strands of DNA to form that complex helical structure. So this and this are the bases from different strands of the DNA, which tends to bond to form this helical structure. So now let's come to the base pairing and the complementary group. In a DNA molecule, you have the same amount of adenine pairing with thymine, because when you look at this one, This one is different nitrogenous base. This one is different. This one is different from this. This one is different from this. It means there is a rule that governs the bonding system initiated in the nitrogenous base to form that double-stranded DNA. In the DNA molecule, keep in mind that adenines always pair with thymines. So adenine, which is a purine, a double ring, always pair with thymine, which is a single ring. And also the guanine pair with the cytosine, which is this one is a purine and this one is a pyrimidine. So keep this rule in your mind. If you have this, then it means that when you take every DNA molecule, the number of adenines and the thymines are the same, because the adenine always pair with thymine. So the number of adenines and thymines in every DNA molecule is the same. In the same way, the number of the guanines and the cytosines are also equal, because the guanines always pair with the cytosines in every DNA molecule. So let's take some of these mathematical examples and see how we can use this rule to solve them. So supposing we have a particular DNA molecule and we had 60% or 15% of them being the cytosine. The C is 15. So based on this rule, we are saying that the cytosine and the guanines are the same. If we have C of them being 15, then it means that the gene is also 15, of course, because the cytosine and the guanine in every particular DNA is the same. So you have 15 being cytosine, then the guanine also becomes 15. Then the remains of the DNA or the nitrogenous bases becomes the adenines and the thymine. So if you have 15 of them being cytosine, then 15 being guanine, it means the remaining 70 is the adenine and the thymine. Since the adenine and the thymines are also equal, then it means that the 70 divided by 2 is going to give you the number of adenines and the thymines in the DNA molecule. 15 being guanine, 15 being cytosine, then it means that the adenine and the thymines are all 35% because they are all equal. So 15 plus 15 plus 35 plus another 35 make up 100%. So try this example and let's see. You can drop your answer at the comment section after watching the video. Assuming you have a DNA molecule and the number of the guanines are 2000 and we have total number of 12,000 nitrogenous bases in the DNA molecule. What is the percentage of the thymine? This is a very critical question. We have guanines to be 2000 and total number of 12,000 nitrogenous bases in the molecule. What percentage of the bases are thymine? It's very simple. Try it and drop your answer at the comment section. Let me give you the overview. If you have 2000 being guanine, then it means you have the same amount 2000 being cytosine. So 2000 giving you 4000. It means it will be left 8000 shared among the adenine and the thymine. So you have 8000. It means that the 8000 divided by 2 is going to give the number of adenine and thymine. So it's going to be 44. 4000 for each. But what percentage of it is it going to be? So just take the number of thymines, which is 4000, over the total number of the bases, which is 12,000, then multiply it by 100, give the percentage of the thymines in that particular DNA molecule. So this one shows how the bases are paired in a DNA molecule. So when thymine, as we said, bonds or pairs up with the adenine, the thymine pairs up with the adenine due to structural differences. These bases are joined by what we call hydrogen bonds, and the thymines and adenine, due to the structural differences, initiate only 2 hydrogen bonds due to how their structure looks like. In the same way, the cytosine and the guanine initiate 3 hydrogen bonds because of their structural differences. Here we can say that the bond between the cytosine and the guanine is very strong than the one between the thymine and the adenine because of the here being 2 hydrogen bonds and this one being 3 hydrogen bonds. Cytosine and guanine are joined strongly than thymine and adenine because of the 3 hydrogen bonds, which makes them more stronger than the two hydrogen bonds. DNA is complementary to each other. So complementary, it means that every nitrogenous base pairs up with its continuous or complementary nitrogenous base. That makes every DNA molecule complementary. So let's look at this strand of DNA. So this is how a DNA molecule looks like. It has two strands. This is one strand and this is the other strand. Each strand has a backbone. The backbone is composed of the phosphate group and the deoxyribose sugar. The sugars are joined by the phosphodiester bond in the backbone, and this one through the same thing. The bases are paired complementary. So the cytosine of one nucleotide pairs up with the guanine pairs up with cytosine, the thymine pairs up with adenine in that complementary base pairing rule. So this is one polymer composed of several residues of the nucleotide made of the nitrogenous bases, the deoxyribose sugar, and the phosphate. And these sugars are joined by the phosphodiester bond. This strand and these strands come back to form the DNA molecule by the joining of the nitrogenous bases based on the complementary base pairing rule. The guanine and cytosine has three hydrogen bonds, whereas the adenine and thymine has only two hydrogen bonds. That makes the GC a stronger bonding system than the TA. So let's look at this structure. We are saying that the DNA molecule is an antiparallel molecule. It means that when we take one strand runs in opposite direction to the other. When we take every DNA molecule, it has two ends. It has the hydroxy end and the phosphate end. And the phosphate groups are always ended at the fifth carbon of the ribose sugar, whereas the hydroxy group ends at the hydroxy or on the third carbon of the next or the other ribose sugar. So we take this backbone, we have the phosphate on the fifth carbon of this, then hydroxy on the third carbon of the sugar. Because of this ruling from the third carbon to the fifth carbon, we see DNA strands runs in 3 to 5 direction. 3 to 5 direction because of the ending on the third carbon of the sugar, then the ending on the fifth carbon of the sugar. The phosphate is always on the fifth carbon of the sugar, whereas the hydroxy is on the third carbon of the sugar. Due to this orientation, we see DNA molecule runs from 3 to 5 direction. When we take this one, the other one will run from, if here is 5 to 3 direction, this one will be from 3 to 5 direction because here it starts from hydroxy and ends at phosphate. So while this one from down here runs from 3 to 5 direction, this one runs from 5 to 3 direction. Up here, while this one runs from 5 to 3 direction, this one runs from 3 to 5 direction. So we see, based on this, we see DNA molecules is antiparallel and the two strands run in opposite direction. That's how basically DNA molecules look like and how they are paired up to form the complex molecular unit. Don't forget to subscribe after watching so that you keep in touch with every update from this channel. Thanks for watching.